Welcome, everyone. It is my great pleasure to introduce Alejandro from Dow Labs.
Hello, everyone. I'm really excited to talk to you guys today. I have a presentation and, I plan to tell you a little bit about myself, what my job essentially is about, and do a little demo for you guys. It's kind of like a little magic trick. I'm making something called the carbopol gel, and it should be really fun. I'm really excited to be here with you guys. I wish we could have you guys on site. But maybe we can once the pandemic's over, we can have you guys come visit and maybe give you guys a tour and stuff. So thank you guys for having me. I'm really excited to talk to you guys. I wish you guys could come down to our facility and tour the place. We'll probably have to save that for some other time once all this pandemic stuff is over. Hope you're all doing well. I'll get started by telling you a little bit about myself and essentially my journey to becoming a chemist. So I grew up in Bakersfield, California. My first language was Spanish, and I didn't learn English until I was 6 years old. And I actually had a really hard time catching up to the appropriate reading levels and writing levels, and I was in English development classes until the end of middle school. So I had to work really hard to catch up, and eventually, I did a lot of reading, a lot of writing, and I was able to catch up to the appropriate reading levels and writing levels, and I was even able to eventually get into an AP class. So I definitely had trouble early on, kind of with school. As I went along, in high school, I discovered science and I fell in love with it. It was easy. I was good at math, and definitely something that I loved, experiments, seeing demos and things like that. Another thing, living in Bakersfield, it was really easy to get into trouble. So for me, I made it my goal to essentially go to college and try to be a good example for my siblings, so I could be a good role model for them. So what I did was essentially, talk to a counselor, because I didn't know what to do to apply to college. So what I did was I went to my counselor in high school, to his office, and I asked, what do I need to do to get into school? And then I learned about, application deadlines, scholarships, FAFSA, and essentially I applied to different schools and different scholarships, and I was able to get into a lot of schools, and also get a lot of local scholarships. So once I heard back from schools, I decided to go to UC Davis and study pharmaceutical chemistry. Once I got to Davis, it was a little difficult as well, kind of my first year, because I didn't know what to expect, being first generation in my family to go to college. So I didn't know what to expect. I knew I love science, and I needed to kind of figure out how to finish school as well. So my first quarter was actually one of the most difficult, because I failed most of my tests. And that was really shocking to me, because I had done really well in high school. So it wasn't the same as high school. I realized that I needed to study more consistently. I couldn't study the day before and do well. So I had needed to kind of change my game plan and figure out how to improve essentially my study habits. So what I did was I went to a counselor. Colleges have them, and they provide you with a ton of resources. So things like tutoring, they tell you about office hours. They also have workshop classes, and They help you essentially get the help you need so you can do well. When you guys go to school, I highly recommend you guys going out to and reach out to a counselor to kind of set up a game plan so you can succeed. After that, I got the hang of it. I got my study habits down, and I didn't know anyone because I was actually the only person in my school to go to Davis. So it's also important to kind of have a balance between studying and kind of social life, because you'll go crazy if you just study all the time as well. So what I did is I looked for internships, and I looked for organizations that I can help and volunteer and things like that. So some of the things I did was I volunteered for Relay for Life. Relay for Life is a organization that fundraises money for cancer research. I gained a lot of leadership experience and organization skills that I still use today. At the same time, I was interning at Rite Aid in a pharmacy, and I wanted to get familiar with all the drugs that are out in the market. At the time, I thought pharmacists were the people that made the drugs and then would give them to you. However, that wasn't the thing. I found out that most of this stuff is already made, and essentially they just fill the script and hand it to people. So I learned, that pharmaceutical companies are the source of manufacturing and developing drugs. So I wanted to do that once I graduated. So my senior year, probably 6 months before graduation, campuses usually have a career fair. And at the career fair, companies from all over the US essentially come and are looking to hire new graduates. Before I went, I did go to the Career Center. The Career Center gives you a ton of resources as well. They help you make your resume, they do coaching, and they prepare you to essentially go to the career fair and talk to these companies. It's super important to know, do your research, be prepared, have good questions. It's kind of like the first impression matters. At the career fair, you have about a minute, 2 minutes to make a good impression, so they can put your resume aside and contact you later. So I highly recommend you guys going to the Career Center and working on your resumes, coaching for interviews and things like that. So after going to the Career Center, I applied to multiple companies, and within 2 days, I heard back from Robert Mondavi Winery. It's located in Napa, and it was a lab position to essentially help the manufacturing of wine. We did a lot of testing, wet chemistry. I learned how wine is made, and it was a great experience, and I really enjoyed it. It's a great place to work at. As you can see in the picture here, you can see the facilities are wonderful. So once I got that job, I actually only was contracted for 6 months, so I needed to figure out the next thing. So I slowly built my skills up to get to kind of the pharmaceutical job I wanted. I didn't get it the first shot. And sometimes this happens, you don't get your dream job right out of college. You have to kind of work up and eventually get there somehow. So working at a winery, alcohol is one of the oldest drugs known, and it was an opportunity that I learned a lot of skills, whether it was lab or just production in general, and fermentation and things like that. Then after those 6 months were done, I got a job at Eurofins. At Eurofins, I tested a lot of dietary supplements, such as proteins, powders, vitamin C, things like that. And my job was to essentially break these products down and bring them back down to the smallest molecule. So a lot of extractions, a lot of wet chemistry, and kind of decomposition of a product, essentially. Kind of going the reverse way. I learned how to use instruments we learned at school, like GCs and HPLCs. HPLC stands for high performance liquid chromatography. These instruments are very valuable and very helpful because they help you kind of quantify how much of a compound is in a product. Also, once I had that experience, I worked there for about a year and 9 months. I finally felt like I had enough experience to go and get, a pharmaceutical job. And I always wanted to make stuff. So I was breaking things down from product to analyte, essentially. But my dream was to work at a pharmaceutical company and, maybe one day be watching TV and see a commercial of a product that I helped make or develop. And I found out that at Dow Development Labs here in Petaluma, they help other companies develop topical products. So I applied and I got the job. And I'll tell you a little bit about what we do here at Dow Development Labs. We are a contract laboratory located in Petaluma. We work on specifically topical drugs and develop, test, and manufacture them. Our clients range from early startups to large pharmaceutical companies. Here's a picture of our R&D laboratory. The big picture of our work is to help clients develop topical drugs for different skin conditions. We make things like gels, creams, ointments, solutions, and suspensions for new chemical entities, approved drugs, small molecules, and proteins. All these products essentially help treat conditions such as acne, psoriasis, atopic dermatitis, and things like that. So essentially our ultimate goal is to help companies make products that are safe and effective. However, there's a lot of science that has to go into these studies to bring a product out to the market. So I'll go through kind of a little bit of a summary of how a project starts and where it can end and for approval. So usually it starts with pre-formulation. Pre-formulation is usually we get the drug, and you can't just put powder drug on you. That doesn't help. It has to be essentially put in a vehicle, and that's what we do on site, essentially to help develop a vehicle to get it through the skin. And it all starts with solubility. So we want to make sure that we can dissolve the drug, or we want to know if we cannot dissolve the drug to essentially put it in the right system, whether it's a dissolved system or a dispersed system. And this study essentially helps us gather that information and gives us a good starting point in development. Once we have that information, we start developing prototypes. We go into the lab and we use mixers and hot plates. It's a lot like cooking. It's really fun. And we make these formulas. So sometimes, some of the prototypes don't work. And here's an image of a cream I tried to make. However, it looked more like cottage cheese that didn't come together. it did not look good at all. And the next one that we tried was successful. And the way we did it was we learned from the old one, and then we were able to build on that and make a successful product. Here it is under the microscope. As you can see, the one on the left looks terrible. There's fluid coming out of here, which is something we don't want to see. What we want to see is kind of this uniform droplets, very homogeneous, very uniform overall. And that's what we want to see when we create prototypes such as a cream. Once we have a couple prototypes, we want to test their stability. The importance of this is to kind of determine a shelf life for that prototype, and also make sure that it doesn't go bad and kind of degrade in a week or two. So usually what we do is we stress the samples at higher heats and higher humidities. We put them in incubators such as these depicted in the slide. Usually these studies last from 3 to 6 months and can allow us to determine a shelf life for the product, but also is useful information in case someone leaves their cream in the car and they leave it there, and we want to make sure it can tolerate high heats and things like that. So stability studies help us determine that. We use our analytical group here a lot when we do those studies. They test, pH, appearance, make sure everything looks the same at different time points. They make sure that the purity of the initial product hasn't degraded, and make sure that the product is still stable. Once all this stability data is gathered, we can move on to the next steps in development, which for us, we help companies. We make essentially GLP batches for animal studies. GLP stands for Good Lab Practices. These batches are usually made in the laboratory, the R&D laboratory, but with a lot of paperwork and a lot of regulations. So here's an image of kind of a batch that we made. It's a 25 kilo batch. This batch was gonna go for animal tox and studies. So what companies have to do is usually send batches out to for animal studies. You want to make sure it's safe before it's put on a person. So a lot of animal studies are done to pharmaceutical products before they go on to human beings, and they're very expensive, very elaborate, and also just give a lot of information when it comes to safety. Once all the animal tox studies come back and their companies are ready to proceed, we also help make GMP batches for phase 1, phase 2 trials. We do not do GMP work for phase 3 studies. So I'll tell you a little bit about the batches that entail GMP. So we manufacture these batches in controlled rooms. You can see here, these are clean rooms right here. They're highly controlled environments, and all batches that are made in these rooms will be put on people now for development studies. So essentially, once we make a GLP batch, GMP batch, those batches can be used to go into clinical studies, and the first one is phase 1. Phase 1 helps test the safety of the product on healthy people. So essentially what the FDA wants you to do is to put or prove that this product is safe to put on healthy people before you put it on someone that has the condition. These studies are also very expensive and very long. Usually the study groups are pretty small, and they essentially inform and make sure that the product is safe before you go on to put it on sick people. Phase 2 tests the safety on patients that have the indication. So now you're putting the product on people that are sick, and now you want to prove that it is safe, it doesn't irritate them, things like that. And you also test different strengths and determine proper doses and things like that. And then finally, phase 3 testing, you test for efficacy. So essentially, you want to make sure that the drug gives you the desired response. So in these studies, you usually have a placebo and a active, and essentially what needs to be proven is that the active outperforms the placebo. So once all of these studies are complete, then you're essentially ready to go to the FDA and get an approval. So you submit an application with all the data you've collected. If the product is approved, then essentially you can treat patients right away and then go to market. If the FDA rejects it, they need to tell you why, and then you have to go back and correct deficiencies, and that takes more time and more money. And that's it for. So essentially, carbapol is a polymer. It is used a lot in gels and also used as a suspending agent. It is also used to stabilize a lot of creams and emulsions. It's a great material. So what I did yesterday was I dispersed the carbapol in water. And when you neutralize it, something very interesting happens. So have you guys ever wondered, like, why the little, in your cleansers, why little beads essentially are suspended and gravity doesn't bring them all the way down? So carbapol is a suspending agent and is the material that allows that to happen. So I want to kind of just demonstrate that for you, how great of a suspending agent it is. So I have kind of just glitter, and I am going to add some in there. So you can see that everything is kind of settled at the bottom, like you can see right there. So carbapol is a great suspending agent. It kind of freezes everything. It's like taking a picture. So sometimes some cleansers that you see in the store have the little beads, and this material allows that for that to happen. And the next thing I'll do is add some base. So by adding base, what we did is now cross-link the polymer, and you can see it's holding the entire thing. You can see when it was fluid, now it's not. It became a gel. And it's nice and thick, and you can see that the glitter is suspended. So this is essentially how some cleansers stay suspended, and gravity won't bring these little particles down. And you can also see it was hazy, but once we cross-linked it, it turned clear. And that's the magic of Carbaball. One more thing that this material, a unique feature of this material is that it's not ion tolerant, so it doesn't do very well with salts. So I have, when you add salt to it, I'll demonstrate. So here's just some gel. So I'm gonna add some salt to it. And it goes back where it was initially. It becomes a liquid, and everything starts flowing down to the bottom. This feature is pretty cool because when you have a gel and it touches your skin, it actually helps smooth it in and spread it out. So that's one cool feature of Carbaball, and helps a lot. And essentially, that's the little demo, and I had a lot of fun showing you guys. Sorry about the technical difficulties at the beginning, and thank you guys very much.
Great, thank you. That was such a cool demo, Alejandro. We've got some great questions in the chat, so I'm going to start reading them out. First question is from Seraphim, and he asks, do you test how long the drug remains effective in the body, and how many intakes must one take per month in order for the drug to remain effective?
So we do not do any of that testing. We make the batches that go to the clinic, and clinicians and doctors end up doing those studies to find the appropriate doses, and at the same time kind of see how many times per day you need to do it. So we don't do those studies here, but we do just manufacture those batches that get used for those studies.
Great. And then Carrie asks, what would you say your favorite part of the process is?
So I think my favorite part of the process of making stuff is when all the chemistry just happens. So in my demo, you can see it started off all kind of just cloudy, it looks terrible, and then you add one ingredient and then kind of the reaction happens. And things thicken up, things go from a terrible state to something, a product essentially. And that's probably my favorite part of the manufacturing process.
And Sophia asks, around how many trials do you take to make a drug? What does your typical workday look like?
So usually we don't do the trials here. I think my day usually is we supply a lot of the batches that are used for these studies. So my typical day is I come in, I check my email, I have a morning huddle with my boss, and we kind of see what we're going to do for the day. Once we determine that, I go to my desk and I write my experiment. There's a lot of documentation that goes, so I have to set up my notebook and set up the lab to execute my experiment. So for example, if I go and I am making a batch, a prototype, I set everything up, and then I make the batch and I write all my observations. Once I have everything written and everything's complete, I clean up and then wrap essentially all the little small ends to kind of just go home, and that's how my typical day goes. In terms of how many trials you need to approve a drug, there's a lot of work. I just kind of summarized it a little bit, but there's animal tox studies, there is human phase 1 trials, human phase 2 trials, and human phase 3 trials.
And then Izzy asks, how often do drugs have unexpected side effects?
So drugs always have side effects. Like if you see any commercial, you'll see that there's always a big list at the very end that they say that the drug has side effects, essentially like a list of side effects. And for the most part, every drug has a side effect. And I haven't heard of any one that does not have one.
Carrie wonders, do you encounter many, if any, ethical problems in your work?
Can you repeat the question? Sorry.
Do you encounter any ethical problems in your work?
No, not really. Like in terms of what we do is just chemistry. We don't deal with anything that's unethical. We follow the rules. We keep proper documentation and things like that. So no.
Jake is wondering, are there any new, interesting, or promising advancements in your field?
There's always products that people want to make. I can't really disclose like anyone specifically because we have confidentiality disclosures, but yeah, there's some stuff that's really interesting and could really help a lot of people if they do get approved.
Well, and Aaliyah is wondering, have you created your own formula and what was it used for?
So I've tried many formulas actually. A lot of them have failed. It's also part of the job in science. There's a lot of failure to it. There hasn't been anything that has gone all the way. One day I hope that does happen. And yeah, that's pretty much it.
Great. How long does the creating and testing process usually take from start to finish?
So it all depends. It can take up to 10 years sometimes, but it all depends on what stage and what you're kind of doing. If you already have, let's say a company wants to develop a drug that is already known, that cuts the time a lot. But if it's a new chemical entity, then it does take a long time, like about 10 years.
All right. Oh, and someone had a question about the formula for N95 masks.
Formula for N95, what do you mean?
If you know anything about how those are created.
No, I do not know how those are created.
Great. And did you mention how much it costs to develop a drug?
No, it's really expensive. Again, it depends on what you know and what stage you begin and end. So the number that usually gets kind of thrown around is about between $50 million to $1 billion. So that's a wide gap. So it all depends on kind of what stage you're in and how much information you already have on the product. So it's very expensive. And it's kind of one of the reasons why, you know, your cream of acne cream is so expensive as well.
Okay. Unless anyone has any more questions, just want to say thank you so much, Alejandro. And at this point, we have one more. Do you test how long the drug remains effective in the body, and how many? Oh, yeah. Do you test how long the drug remains effective in the body?
No, those are usually for clinical trials. We just make, we test, and kind of supply the materials for those studies.
Got it. Okay, so at this point, I think we are ready for the worksheet.
Okay, sweet.
Yeah. So are you going to have time to stick around a little bit, or are we saying
Yeah.
goodbye?
I can definitely stick around.
Okay, so we are going to make breakout groups. I think we may need to make Rosalie the host again.
Okay. Do I need to do something?
I think Rosalie can do that. And then we'll have everybody, there's going to be breakout groups, and you'll have the option to join in just a minute. And let me go ahead. I'm going to put the worksheet that we're doing. It's on our class site, but I'm going to put it in the chat for everybody. Okay, there it is. So if you guys want to click on that worksheet, that's the worksheet we're going to be doing today. We've got Daniel and Fiona, our TAs, that are going to be helping in the breakout rooms. And so what are you working on today?
Today, solubility, actually. So that's the experiment I'm doing today. So I have a drug, and I'm essentially looking for solvents to dissolve that API, and so that's what I'm working on today. Things are shaking in the lab as we speak.
Cool. Do you have interns in your lab?
We do. We have had interns before. I don't know with the circumstances right now what those are, but yeah, we do have intern positions that we have students come in and essentially work and kind of see and expose them to what we do here and stuff like that.
What would you say is the other than chemistry, what's the most important class in high school if you wanted to become a chemist?
I would say biology. They kind of go hand in hand. Like chemistry is, you know, the building blocks of biology, and kind of they build upon each other. So physics, you know, first it's physics, then chemistry, then biology, and they kind of all interconnect. So I would say biology is probably a really important course, and allows you to apply chemistry in real life, essentially.
Cool.